everyone can hear me, and thank you for all being here. I'm excited to tell you about our lab's most recent data indicating that chemo-residual tumor cells of the triple-negative breast cancer subtype are actually more invasive and metastatic than the untreated tumor. So I included this slide first to remind everyone that breast cancers are not all the same. They come in different subtypes. These subtypes differ in the expression of hormonal receptors like estrogen receptor and progesterone, as well as the EGFR family member HER2. So today I'm going to be focusing on this subtype called triple negative. It's called triple negative because it lacks the hormonal receptors and HER2 expression. So there are no targeted therapies available for this subtype of cancer. Even though it only represents about 15 to 20% of all breast cancers, it's particularly needed to study why these cancers are so aggressive and untreatable. So like I said, the triple-negative breast cancers currently have no targeted therapies available. Although many of the patients do respond to chemotherapy, the chemotherapy response is incomplete in about 60% of the cases. So the residual cells that do remain in these patients after chemotherapy treatment frequently result in a future tumor recurrence. And the frightening statistic that I like to include in my talk is that a third of these patients that have an incomplete chemotherapy response will die within 3 years after treatment. So a lot of need to understand chemo-residual disease. So one model of chemoresistance indicates that tumors aren't homogeneous. They consist of both chemosensitive cells and chemoresistant cells. So when we treat with chemotherapy, although there is apparent tumor regression, there are chemoresidual cells. And if given time, these chemoresidual tumor cells eventually form tumor recurrence. So in my mind, I think it's really important to study these chemoresidual tumor cells in patients in order to identify novel signaling pathways that we may target to combine with chemotherapy. So my lab has invested a significant amount of time studying tumor heterogeneity in this particular subtype of breast cancer. And what we wanted to know was how to enrich for these rare therapy-resistant cells in order to study their unique signaling properties. So as an outline of my talk today, I'll first describe our short-term chemotherapy treatment model that we've optimized to study signaling pathways in chemoresidual triple-negative tumor cells. And then I'll show you evidence that these chemoresidual tumor cells from our model exhibit highly invasive behavior associated with the expression of a novel adhesion molecule. So just to put our work in a little bit of context, I wanted to talk about how people have classically studied chemoresistance in cancer. And this involves in vitro studies where we expose tumor cells to chemotherapy for up to a year at very high escalating doses. And what we do is we drive mutations that confer chemoresistance. And I think the question is, how relevant are these resistance models to patients? I think this is a very controversial topic. So we wanted to employ an acute chemotherapy treatment model in order to study the chemoresidual cells that may occur in the patients. And I'm showing the primary slide of this. Actually, I think I skipped over one. So what we do is we treat these, what we consider to be heterogeneous tumors, with acute chemotherapy treatment of 2 days. We get our chemoresidual cells and then remove the chemotherapy. So we get regrowth of multi-drug resistant cells that we can study unique signaling in. So this is showing one cell line that we study. It's a triple-negative cell line called SUM159. We treat for 2 days with a high but physiologic dose of an anthracycline or a taxane at doses that are achieved in the patients. And on day 2, we remove the chemotherapy. Now what we find is that even after we remove the chemotherapy, there's significant death over about a week. And on day 7, we find a very small number of chemoresidual cells. And most people would just toss these plates because there's so few cells it's hard to study. But we were interested if we just let these cells sit around without chemotherapy, what would happen? And what we found is after about 7 more days, we got the growth of these colonies. And the cells in the colonies were multi-drug resistant. So today's talk is going to focus on characterizing the signaling in these chemo-residual cells. So at this point in the talk, I wanted to remind everybody that it's thought that although chemotherapy does target the tumor cells and eliminate the bulk, there are residual cells that are thought to be invasive and to gain access to the vasculature, allowing them to form recurrent tumors at distant sites. So what we were interested in knowing was whether in our model the chemo-residual tumor cells would exhibit invasive behavior. And the first assay we used to measure this is called a trans-well invasion assay. So in this assay, we're coating our membrane with Matrigel. We're adding our cells to the top chamber and studying their ability to penetrate the Matrigel and chemotax towards a conditioned medium. So what we do is we stain the cells at the bottom of the membrane in order to determine the number that are invasive. And without exception, when we've put our chemo-residual cells into this assay, they're significantly more invasive than the parental cells. We're showing that here in two different cell lines. Quantification of the invasion is shown below, and you can see it's about, the chemo-residual cells show about a twofold increase in invasive potential in this assay. But we didn't want to limit our studies to in vitro, so we wanted to look at what might happen in a tail vein injection of mice. So in this study, we've taken 10 mice and injected them with either parental cells or the chemo-residual cells from this model. And we were struck that when we looked for lung colonization in these mice, the chemo-residual cells were much more effective at forming lung colonies than the parental cells. And in addition, if we counted the number of lung colonies per mouse, we saw a lot more in the mice injected with the chemo-residual cells than we did in the mice injected with the parental cells. So this suggests that these chemo-residual cells have metastatic properties much higher than the parental cells do. We've also looked at this by bioluminescence because we had transfected our cell populations with luciferase, which allowed us to track the movement of the cells in the mice. And what we consistently saw was luminescent cells in the lung of mice injected with the chemo-residual cells, but not in the lung of mice injected with the parental untreated tumor cells. So a colleague of ours had published, David Coleman, had published a paper indicating that an adhesion molecule called N-cadherin has a proform, and then on tumor cells, this proform drives invasion. So we were interested in looking at this molecule in our model. And just as a little bit of background, this is a schematic of how the N-cadherin adhesion molecule gets processed in cells. It's synthesized as a proform, but this pro-domain gets cleaved by proteases in the Golgi apparatus. This results in the transportation of this mature form to the plasma membrane. So N-cadherin adhesion function it requires the pro-domain cleavage. But what Dr. Coleman's group showed was that tumor cells have two flavors of N-cadherin on the cell surface. They have the mature form, but they also have the proform that has this extra domain on it. So what this group showed is that in pre-malignant cells are highly adhesive because they have the mature form only of N-cadherin on their cell surface. But when he looked at low-grade and high-grade tumors, he saw a mix of the proform and the mature form, and this mix seemed to drive invasive behavior of the cells. When we looked in our model, we saw that the chemo-residual cells had significantly more pro-N-cadherin than the parental cells. This was true of multiple triple-negative cell lines. And we did flow cytometry to confirm this on the cell surface. So what we saw was that the chemo-residual cells expressed a lot more pro-N-cadherin on the surface than the parental cells. This is in contrast with just the mature form of N-cadherin, which was equal in the two populations. But the ultimate job we had to face next was to really show a correlation between this pro-N-cadherin expression in our chemo-residual cells and an invasive phenotype. So we went back to our heterogeneity model, and it was our hypothesis that these chemo-resistant tumor cells that pre-exist in the original tumor are the ones that express the pro-N-cadherin and are highly invasive. So we performed a sorting strategy to isolate the pro-N-cadherin positive and the pro-N-cadherin negative cells. And we took these populations and put them into the Transwell assay that I described earlier. And what you can see is that the pro-N-cadherin positive cells that have been sorted from the original tumor are more invasive than the pro-N-cadherin negative, with quantification shown on the right. So this is a stem cell meeting, and whenever I give this talk, the first question I am asked is, well, are your chemo-residual cells cancer stem cells, which is a very controversial field. So just to define what dogma says about cancer stem cells, we need to show that these cells exhibit the ability to grow in the absence of serum as non-adherent self-renewing spheres, or in our case, mammospheres. And we also need to show that these cells show increased tumor initiating activity in a xenograft model. To put our work in context with the rest of the literature, other groups have already shown that if you treat triple-negative breast cancers with chemotherapy for 4 continuous days and then take the cells that remain, they're highly spherogenic, suggesting they're cancer stem-like. And this has been shown by 2 groups, both Semenza's group and Artiega's group. But I want to point out the difference, again, between the previous models that have been described in this model that we've developed in the lab. In this model, we're using acute chemotherapy treatment, removing the chemotherapy, and seeing what grows out. So now we had to face this question. In our model, are the chemo-residual tumor cells cancer stem-like? And the bottom line is no, they are not. And in fact, the parental cells, the untreated triple-negative tumor cells, show much higher spherogenic activity than the chemo-residual cells do. To really nail this down, we also did a xenograft study injecting different doses of the parental and chemo-residual tumor cells into the mammary fat pad of immunodeficient mice. And here we're looking at the percent of mice with tumors less than 100 millimeters cubed over time. And the bottom line is that with no injection cell dose, did the chemo-residual cells show increased tumorigenicity compared to the parental. So it doesn't appear that the chemo-residual cells have higher tumor-initiating activity than the parental cells, indicating that they're not stem-like, contrary to previous models. So as a summary, we've shown that chemo-residual tumor cells of the triple-negative subtype, evolving from our short-term chemotherapy treatment model, demonstrate increased invasive behavior, increased spontaneous metastasis, but do not demonstrate cancer stem-like cell behaviors. I wanted to talk about our work in the context of a previously published work by Dana et al., in which they primed a mouse with chemotherapy before giving the tumors. And they asked the question, does the chemotherapy create a metastatic niche that allows the tumor cells to metastasize better? And quite strikingly, if they did this pre-treatment of the mice with the chemotherapy of different classes, paclitaxel, cisplatin, they saw that when they did inject the tumor cells, they were more metastatic. So pretty interesting that you're somehow affecting the host by chemotherapy treatment to create a metastatic niche that allows the tumor cells to metastasize better. Now, our work is distinct from this because it's showing that the chemotherapy treatment of the tumor cells directly impacts them by selecting for a metastatic subtype. And one study we're really interested in doing is determining whether there's additive effects of the chemotherapy driving metastasis in the tumor cell, but also the chemotherapy priming the host environment to promote metastasis. So what are the implications of our studies for triple-negative breast cancer treatment? At face value, chemotherapy can cause tumor regression, but in the triple-negative breast cancer case, we also think that chemotherapy may enrich for chemo-residual tumor cells with increased metastatic potential. So I think there's a real urgent need to target these chemo-residual tumor cells to prevent tumor recurrence. And we've identified several unique signaling pathways in the chemo-residual tumor cells that we'd like to target in combination with chemotherapy to see if in a clinical trial it might benefit the women in terms of survival. So just a quick acknowledgement slide. The individuals in my group that were central to this work, Shendo Lee, Matthew Lies, Maggie Kennedy, Sturgis Payne, and Kelly Kennedy, and our collaborators, Eric Nelson, Donald McDonald, and Sal Pizzo. I'll be happy to take any questions.
Any questions?
Oh, okay. It's interesting, very interesting results. Congratulations. I'm just concerned about the results where you get increased metastasis using the tail vein injection, but then you don't get increased tumor initiation. When you do tail vein injection, you are skipping so many steps of metastasis.
Absolutely, yes.
And then when you do tumor initiation, that is when you are doing, I'm assuming you are implanting the tumor into the mammary fat pad, right?
Yes, that's right.
So if they don't have an increased potential for tumor initiation, but then as compared to the bulk tumor, because we know that the bulk tumor definitely is more heterogeneous, and the more dividing cells are the ones that are responding to chemotherapy. So the ones that are not responding to chemotherapy are assumed to be either more dormant or they were not dividing at the time of chemotherapy. So if it is not more tumor initiating as compared to the bulk tumor, then the results about the metastasis becomes a bit skeptical. Mm-hmm. Because you have, if you did like implanted both, and then you look for metastasis, because you can look for metastasis when you implant both the bulk and the residual tumor in the mammary fat pad. And then you wait for a couple of months, and then you look for metastasis. Then you can find metastasis in the liver, in the lungs. That is the best way you'll be able to compare metastasis between the bulk tumor and the tumor and the residual cells. Then you'll be able to say that that is more metastatic than the previous one. But then if you do tail vein injection and you say that one is more metastatic, maybe there are so many factors that might influence that. And so
it's an excellent point. Excellent point. The spontaneous metastasis assay is very artificial. We have looked for metastases in our mammary when we injected the cells directly into the mammary gland. We did not see they formed tumors better. We waited for a period to look for metastases, but the problem with that mammary gland injection model is that you don't get metastases unless you remove the primary tumor. And that was true in our case. None of the mice developed metastases. So right now we're repeating the study. We're removing the primary tumor after it forms and then looking for lung metastases, and that will be a much more physiologic model. You're correct.
James.
Have you looked to see if the cells, the resistant cells that are derived under the acute treatment are different in ways from the ones that are sort of typically derived in resistant studies?
So
Could you say a little bit about how they differ?
I'd love to. We've done microarray study on the chemo-residual cells and identified some very unique molecules. We're currently writing up a paper which shows that there's a nuclear growth factor in the cells that's never been described, nuclear fibroblast growth factor. It drives DNA repair, and this hasn't been described in the long-term chemoresistance models. So yes, that's one avenue we're studying. We're also studying IL-6 in this population, which seems to be upregulated.
And your work brings up another point in terms of looking for cancer drugs, because currently in industry, as far as I know, there's a lot of screening, early screening against the NCI 60 line, which are all tumors that grow very well, more likely to be like cancer stem cells than the things that are actually appearing in patients after acute treatments. So is anyone, or are you thinking about developing a panel of acute resistant cells that might be used for early screening for cancer drugs?
A panel? What do you mean by a panel?
So lots of different types of tumors, treated acutely with lots of different types of chemotherapy agents that are already out there.
So other than the triple-negative subtype, you're saying? Yeah, it's just something we haven't gotten to yet, but I don't know how universal this is. I don't know if it's specific to the triple-negative subtype. We're also studying ovarian cancers, which seem to have very similar biology to triple-negative. Would be a great study. We just haven't had the time to do it yet.
I was going to ask a related question.
A quick one, because we really need to keep the time.
Whether there are any cell lines established from these, the triple resistant tumors Are there any cell lines in existence which have been established from these patients?
For triple negative, well, the SUM159 example I gave is an example. There are numerous ones, and that's what we're starting with, and we're selecting for the chemo-resistant ones. Okay. Okay, thanks very much.
Right. Thank you very much. You have a certificate here.
Thank you.
Thank you. Thank you very much.